The mitochondria is the powerhouse of the cell. This is a phrase that is often drilled into students' heads. The mitochondria has many roles within the cell, including metabolism, cell death, and most importantly, energy production within the cell. Although the mitochondria contain their own DNA, only about 1% of the proteins needed for proper function of this organelle are contained in mitochondrial DNA. In order to maintain proper function within the mitochondria, there is a special stress response pathway known as the unfolded protein response, UPR. This is a mitochondrial nucleus response pathway that results from the accumulation of misfolded or unfolded proteins causing cellular stress.
These unfolded proteins are damaged beyond repair by chaperonins. Chaperonins are needed to maintain quality control within the mitochondria. When they sense an accumulation of misfolded or unfolded proteins, they prevent transport of proteins into the mitochondria. Through studies with the common model organism C. elegans, researchers have found that B-zip transcription factor, ATFS1, to be integral in the UPR response. ATFS1 affects the attenuation of oxidative phosphorylation by having sorting signals for both mitochondrial and nuclear targets.
This allows ATFS1 to be involved in the UPR pathway. Under non-stressful conditions, ATFS1 is sent to the mitochondria to be broken down. But when in a stress environment, the mitochondria will not allow transport of this transcription factor into the mitochondria, causing it to build up in the cytosol. At a certain point of accumulation within the cytosol, it is transported into the nucleus due to the nuclear localization signal. Once in the nucleus, ATFS1 activates a number of genes in the cell response pathway. These genes include chaperonin and protease genes, as well as genes involved in mitochondrial import to allow the mitochondria to return to homeostasis. Similar response pathways have been observed in mammalian cells as well, except the genes involved are different. The three genes involved are CHOP, ATF4, and ATF5. CHOP is a transcription factor known for mediating apoptotic cell death. ATF5 is associated with mitochondrial function, similarly to ATFS1, but is the mammalian analog. ATF4 is also thought to be associated, but its function is unknown.
An experiment done in the model organism C. elegans has shown that the UPR is regulated by ATFS1 by inducing mitochondrial chaperones and proteases. Researchers then wanted to determine if the ATF4 or ATF5 transcription factors found in mammals have a similar process to ATFS1 in worms. The experiment tested ATF4 and ATF5 expression in worms lacking ATFS1. It was found that when expressed, ATF5 localizes to the mitochondria in the absence of stress, but when stress is present, UPR is upregulated in an ATF5-dependent manner. Similarly to ATFS1, ATF5 regulates chaperonin and protease transcription when the mitochondria is experiencing stress. ATF5 has a known role in anti-apoptosis. ATF5 induced BCL2, an integral outlier mitochondrial membrane protein that blocks apoptotic death in some cells, promotes the survival of many cancer cells. In addition to cancer cell survival, further research looks promising in providing improvements in cancer treatment via manipulation and inhibition of the UPR pathway to induce tumor suppression.
Thanks for watching!